Hello, my name is Jeff Saucerman, and I'm a professor of biomedical engineering and cardiovascular medicine at the University of Virginia. I lead the cardiac systems biology group. So the goal of our lab is to identify the molecular networks and control points that decide how the heart responds to stress, with the translational objective to use that knowledge to identify drugs that might generate cardiac repair or regeneration. Many of our projects involve some sort of biochemical or mechanical stress that's associated with heart failure, and looking at how the heart decides between many different options, hypertrophy, fibrosis, death, and proliferation, how it should remodel in response to those stresses. We do that in both using experimental and computational methods. And so on the left, just an example of computational models that piece together little bits of what's known into an integrative framework with which we can predict the outcomes of experiments. And the other way, when we don't have that type of knowledge, using high throughput experiments and quantitative experiments to infer the molecular networks. So when I have a trainee join our lab, whether that's undergraduate, graduate, or postdoctoral, I really commit to mentoring them for their career because their success is my success. I do that through what I call a progressive mentoring style, where we identify initial projects that are well suited towards their existing techniques and would likely demonstrate success, whether that's conference presentations or papers on a short time frame. And from there, once they've demonstrated that success and gain confidence, then we kind of open things up more and more over time, where their kind of later projects are mostly sort of self-guided, that might often be something that's really unrelated to our existing or past studies. And so there's a lot of collaboration both between the lab members and between different labs that I'll mention. And then all of my trainees have some sort of combination of experimental and computational experience, where usually it's kind of more weighted on one side or other based on their career goals and background. So over time, we've trained a number of people that have gone on to a lot of great directions. These are the current PhD students in the lab, and you can see that they've done very well at getting recognized for their projects underway. AHA, NIH, and NSF type fellowships. On the left are some of our collaborators, and then on the right shows some examples of alumni from our lab. And the main point here is that our alumni have gone in a lot of cool different directions, such as going into faculty positions, medical residencies, postdoctoral positions, and a variety of industry positions, mostly in big pharma. So I'll give you just a few kind of research highlights in different areas of our lab. The first one is in cardiac regeneration. And this is a recent study that we did in collaboration with Benoit Bruno, and where they're looking at if you take ES-derived cardiomyocytes and you knock out this gene Brahma, then it completely eliminates cardiac differentiation. And in fact, they found that knocking out Brahma doesn't take it just any cell type. It really rewires towards a neural path, which is really surprising because these cells were already at the mesoderm stage before they had the effects of Brahma. And so, they did a lot of experiments in this study, and Ali Khalili-Mehbadi did the computational models where he found that on a normal trajectory, you follow this path down like this to end up in a high GATA4 state that's associated with cardiac differentiation. If you knock out Brahma, it predicted not only does it end up in a neural potential cell, but it has remaining differential potential. And interestingly, we identified ways that you could rescue this phenotype, for example, with high BMP4. So overall, with some additional bifurcation analysis, this revealed that there's actually cardiac differentiation lies on a one-way toggle switch. And Brahma is not the switch itself, but it guides to make sure that these cardiomyocytes stay on track on this switch. The next example I want to give is more of an application sort of direction, more industry relevant. So in collaboration with AstraZeneca, we've been screening iPS-derived cardiomyocytes and identified a number of different compounds that induce a proliferation of these cells. And so these are some examples here where we identified a number of clinically used L-type calcium channel inhibitors that indeed induce proliferation. And a key aspect here, Laura right here developed very quantitative live cell assays where we could track the actual proliferation, not just cell cycle markers over time. And that was really critical for identifying the best compounds. And now that we've identified a number of compounds, it's of course very of high interest to identify how they work. And so to do that, we've done a variety of RNA sequencing and proteomics experiments to identify and infer the molecular pathways that might be involved. In complementary studies, Brianna Harris in the lab has developed the first computational model of cardiomyocyte proliferation. And so as you can see here, it identifies a number of different like hormonal or growth factor stimuli, such as FGF, neuregulin, IGF, oxygen, that impinge on cardiomyocyte proliferation. It filters down into some key factors like P21, the cyclins, GSK3 beta, things like that, down to not just DNA replication, which is what most markers show, but also making predictions about regulation of mitosis and bionucleation and cytokinesis. And so one of the most exciting findings from this is that the model predicts that a stimulation of YAP, well, it's already known that it's very well known that it induces proliferation. It's not clear how that happens. And so the model predicted that particular PI3 kinase and c-Myc inhibition would reduce the amount of proliferation induced by YAP, this TT10 drug that induces YAP activation. And so consistent with these experiments, consistent with these simulations, our cell-based experiments validated this interesting connection from the Hippo module to c-Myc expression. And especially for c-Myc, we validated that against experimental data in vivo with a YAP mutant mouse. So kind of noting some of the in vivo directions, now we're very interested in taking a number of these drugs and genes forward in vivo in mouse studies. And so this is an example where, in collaboration with Matt Wolf, a really awesome physician scientist and cardiologist at UVA, we're developing automated image analysis methods that we could very quantitatively measure cardiomyocyte proliferation in vivo. So the next area of our lab is in cardiac hypertrophy. And so this is just a highlight of example from Ana Estrada from the Holmes lab that we collaborated with on this. So we had previously modeled the cardiomyocyte hypertrophy network. And what Ana did was she integrated that into multi-scale mechanical models of the heart. So now we can predict how molecular perturbations affect organ scale growth. One example of that has been Kyoko Yoshida's work, also in collaboration as was in the Holmes lab, where she's developing models of some of the molecular pathways that regulate hypertrophy with pregnancy, and validating that against a lot of like in vitro data, and then making predictions about in vivo pregnancy dynamics and how that would be regulated by molecular perturbations. And then finally, in the hypertrophy area, our graduate student, Taylor Egertson, has developed a method to integrate our hypertrophy network models with drug bank, a drug target database, so we can identify all of the FDA and investigational new drugs that might impinge on this pathway. And just a highlight of one example, he found that TGF beta induced hypertrophy is suppressed by midostaurin, a PKC inhibitor that also has other targets and is used clinically for AML. In contrast, we found that midostaurin does not block norepinephrine-induced hypertrophy, which is consistent with our model predictions. And so here's just an example for TGF beta. The model predicts that midostaurin blocks TGF beta-induced hypertrophy, and that also happens in these rat cardiomyocytes. So the final area I thought I'd present today is on cardiac fibrosis. And so Angela Ziegler developed this first large-scale fibroblast network model. And as you can see here, we can predict dynamics on this network. In this case, the dynamics of what would happen in a fibroblast post-myocardial infarction. So these are a method that we use a lot called logic-based differential equations which are really key because they provide dynamics and semi-quantitative predictions, yet are very scalable to these larger sort of networks where the biggest puzzles are outstanding. And so one sort of basic science example of this is she had predicted that stretch-mediated activation of fibroblasts happens not through any direct pathway, but an indirect pathway where there's a autocrine production of latent TGF-beta that gets activated and then signals now through a SMAD3 pathway. And in collaboration with Will Richardson, who is co-advised by me and Jeff Holmes, we found experimentally that in 3D collagen gels, mechanically restrained gels would activate the fibroblasts. You can see them green and spread out. That was blocked by a TGF-beta receptor inhibitor. So this validated the model prediction of a very kind of surprising emergent property. We subsequently took this in a more applied direction, where as was shown with Taylor's project, we found ways to integrate drug target databases where we can screen in these models for many different FDA-approved drugs and whether they might enhance or suppress fibrosis after myocardial infarction. For example, here's an example of arsenic trioxide predicted to enhance fibrosis, as seen in independent studies, or erythritol tetranitrate or other nitrates is predicted to be decreased fibrosis at this intermediate time point, consistent with these studies. Finally, we're not just interested in what's happening inside one cell type, but what happens between different cell types. And so Mukti Chakwale developed this really cool intracellular model that looks at the communication between cardiomyocytes, neutrophils, and monocytes, and also macrophages and fibroblasts. And so here you get the different cell types and how they regulate various cytokines. And then you also have how these cytokines impinge on the different cell types. And so she did a number of cool things in this study. One of them is being able to predict the dynamics of many different cell-specific cytokine expressions and debris and cell behaviors, and then use that to identify some key mechanisms of what we now term inflammation fibrosis coupling. In particular, that IL-1 beta as an inflammatory cytokine recruits monocytes, directs the differentiation of macrophages. Those macrophages, by eating cardiomyocytes, become activated, and macrophage secreted TGF-beta is really key for fibrosis. And that's kind of summarized in this little AI-generated art here. So that's all I have to say for today. And if you're interested in any of these, whether it's a position or just to talk science, feel free to reach out. Thank you.